Hello everyone, in this video we will see how to use GEO2R for data analysis and to analyze different types of plots like volcano plots, scatter plot and box plot, etc. So let's move towards its interface. We'll write GEO2R, then click on this link. Now here you will add the type of the dataset on which you want to work. I will choose this type of dataset, GSE 18842. Now here my dataset basically consists of two types of samples, tumor samples and control samples. If you want to see that which type of dataset this belongs to, you can click on this link and we can see the detail of this dataset. So this dataset is basically related to gene expression analysis of human lung cancer and control samples, published on 1st October 2010. Its purpose is to find out the reliable gene signature to find whether the phenotypic heterogeneity and genetic diversity of lung cancer are correlated with each other or not. Here it basically consists of 91 samples, from which some samples are control and some are tumor samples. As a result, they find out that the KRT15 and PKP1 are highly upregulated genes, and DSG3 gene is highly downregulated in first stage of adenocarcinoma. Basically, it involves 46 tumor samples and 45 control samples, and it has been analyzed with a platform called Affymetrix array. Now, in order to perform data analysis of your own choice, you can download their soft file, minimal file, or series matrix file by simply clicking on these links, and you can basically download them. So let's download. In this way, you can basically download your series matrix file. Now here, soft file is being downloaded. You can also download your series matrix file. But second option is this, like if you don't want to download their files, you can analyze these datasets with the help of GEO2R. So let's click on this button, Analyze with GEO2R. Now the next task is to define the groups. So now here we will define the groups of these samples. Click on this Define Group button and define your sample, control and tumor. Now here we have defined two types of groups. First is control group and other is tumor group. Now let's select the type of samples in each of these groups. We can basically click on this source name and it will divide the control samples and tumor samples in an ascending order. Now if we want to add these samples in the control group, we will first click on the first sample and then we will press Shift button, scroll down. Along with pressing the Shift button, and now click on this control group. Now here we have 46 control samples that we had seen. Now here we have 46 samples in control group. Now let's select the tumor samples. Click on this first sample, then press Shift button. Then scroll down, press Shift button again, click on it, and then select the tumor group. Now we have 91 samples, and out of these 91 samples, 45 are the control samples and 46 are the tumor samples. Now after making the groups, we will move towards Option button. Here we want to normalize our data, so we will click on Yes button. log 2 fold change would be 2, and cutoff value will remain as it is, 0.05. And now here you will select that if you want to use the groups that you had made earlier, click on this button and click on this reanalyze. Okay, so now let's move towards analyzing these graphs. So first of all, this plot is a volcano plot. This is mean difference plot, or it is also called the scatter plot. Now let's understand these plots one by one. So first of all, we have volcano plot, and here this blue color shows that these genes are down-regulated genes, and this red color shows that these genes are up-regulated genes, and this black color shows that these genes are not significant. Now, what do we mean by these down-regulated genes and up-regulated genes? So basically, up-regulated genes are those genes whose expression has been increased as compared to the normal condition, and down-regulated genes are basically those genes whose expression has been decreased as compared to the normal condition. And this black color shows that these genes expression has not been changed as compared to the normal condition, and it remains normal. That's why they are shown by the black color. You can basically click on this explore and download button. And here we can see that like as the genes are upregulated, so their log2 fold change values are in positive, like 2.596, and the ID of this gene symbol is 242931_@. And these genes are down regulated, so their log2 fold change values will be in negative. Here this gene fold change value is minus 3.107. So here we can download these significant genes. If you want to perform further analysis on it, you can use these genes. So here in total we have 1054 gene IDs along with their gene symbols, gene titles, and p-values along with their fold change. So this is the scatter plot. The same color scheme has been used here as like volcano plot. Then the next is UMAP plot. This plot basically shows the groups like control group and tumor group, like how much samples are related to these groups. So there are some samples that are completely related to tumor samples, and there are some samples that are completely related to control samples. So expression is somewhat related to these groups. That's why they are present in these groups. Then we have this Venn diagram that shows that there are 53,622 differentially expressed genes, and we have 1,053 genes that are differentially expressed along with significant, like the threshold that we had used earlier with the p-value less than 0.05 and fold change of 2. This shows that only 1,053 genes are significant along with differentially expressed. This is the box plot. Here this median line shows that it is a straight line, and whenever this median line is straight, it shows that our data is completely normalized and there is no more need to normalize it. There are some other plots, but mostly used plots in our study and our publications is volcano plot, this Venn diagram, and this box plot. Now let's see what is the purpose of these plots, what these plots tells us about. So this volcano plot is basically used to find the genes that are significant along with their larger fold change. And this scatter plot shows that how much genes have been expressed along with their high or low expression. This UMAP plot is And this UMAP plot stands for Uniform Manifold Approximation and Projection Plot. And this is basically a plot that is used to see that how much these samples, like it shows that the sample present in the same group have somewhat similar expression patterns. And these Venn diagrams shows the significant genes along with differentially expressed genes. This box plot is used to show the normalization. Okay, for now, if you want to download your file, you can select the columns of your own choice. You can click gene ID, gene title, gene symbol, adjusted p-value, log fold change, etc. Then click on this set button, download this full table. It's been downloaded. Now let's see how many genes are significant, as we have analyzed in our Venn diagram that there are 1053 genes that are significant according to our threshold that we had used, where p-value is less than 0.05 and fold change value is 2. Now let's apply the filter on this file and see how many genes are significant in our file. It must be 1053. Now here to apply filters, you can go to this data processing button in home tab, click on this filter. Now we will go to this log FC, number filter, custom filter. There are 1053 genes that are significant according to our Venn diagram. So this means that this file is correct. So here we can basically use the gene IDs of these gene symbols, and we can use it in David for pathway enrichment analysis. But in this video, we'll not go to David and we'll analyze the pathway enrichment analysis. We will discuss it in another video. So that's it about this video. If you have any questions, you can drop your questions in the comment section. And thank you for watching.